Hey biology students, this is part 2 of protein synthesis. Part 1 was transcription, where the gene, the genetic coding part of the DNA, has been unzipped. Messenger RNA lines up along its complementary code and bonds to the adjacent RNA molecules, the adjacent RNA nucleotides by covalent bonding, and that strand of messenger RNA leaves the nucleus. So we're now outside the nucleus, and here is a stretch of RNA code, and we are going to go through part 2, which is translation. So let's just say that this picnic table is the ribosome, and I am going to play the role of ribosomal RNA. Now in my last video I was accidentally saying it's going to the ribose, and ribose is part of the word ribosome. It's the sugar that is found in RNA. And so I had to kind of fix that by having the word ribosome show up on my video. So on this one, if the table is the ribosome and I am the ribosomal RNA, what I am is a helper to that molecule in getting the code read. So from the assignment that you just hopefully recently started, you know that we're reading, and I think I had this in the video too, we're reading 3 in a row. So our first codon is going to be AUG. And so this would actually process through the ribosome. It would be moving through the ribosome. And what happens is another kind of RNA, transfer RNA, will match up with an anticodon, so the complement to A, which in this case is going to be not T, because we don't have a T, it will be a U, will complement. And so we'll get 3 in a row complementing, and that transfer RNA is bringing a set of 3 complements, and along with that it is bringing the amino acid that matches that codon code. So here we go. We're going to start with letting you know that Mr. Harrison will be your transfer RNA service. You know, we would have a lot of taxis running, so if you could imagine this were a cell. So we're in the cytoplasm, and maybe the trees are microtubules. I don't know, maybe the support beams are some microfilaments. We've got an endoplasmic reticulum as the river running along. And I don't know, is that a Golgi apparatus in the background or a tent? I can't tell. So we'll just use a little imagination as we socially distance. So this is the social distance version of translation. All right, so this is a reach to the ribosome, and as a ribosomal RNA, I'm going to assist in counting off the 1, 2, 3, and then my first codon from transfer RNA will be coming my way. So here's a very confused and ridiculous Harrison to bring us AUC. And you can say, oh yay, it matches up. So that's not going to go somewhere else. It's the only one place it can go. So it's going to match up there. And then reading the next 3, I need GUC. So what do I need to go with GUC? Complement to G is C. Complement to U is A. Complement to C is G. So
ding, 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 I heard that.
And 3 more. ACA. Can you guys do this for yourself without me saying what goes with this one? Here it comes. All right. So you might notice we did show the complement on this one. We did not tape down the actual nitrogen bases for all of these, but hopefully you get the picture. That would be how they're pairing up. And so just check our work on the initials here and see if we get these right or if Harrison messes one up. So here comes, after the next 3 are read, here comes the transfer RNA with the next 3 for UGC. Good work, Mr. Harrison, good work. And then finally, we didn't have a stop codon in our ensemble at home. So if you would just work with us, we'll just say this is the last one. And for UUG, what would we need as a complement to that? There we go. So these complements again are called anticodons. And so let's see what they brought. These are a variety of amino acids. So these are the monomers of proteins, and these are true to form. Methionine is always the start codon, and you can see they all have this similar backbone, and they actually bond together by way of that part, and you'll see that but they all each have kind of original or different pieces to them as well, making them their own thing. Yeah? Okay. So a combination of many amino acids would result in the primary structure of a protein. So you may be asking, how is it I am supposed to know, how am I going to determine which amino acid goes with UAC? Because you may have noticed you've got some work. You might also be asking, does that dog actually like carrots? And yes, she does. But that's like her binky carrot. It's her security carrot. All right. So for your assignment that has the DNA code written 3 times, and you have some different things you're marking up on that on the 3 different rows, and then at the bottom I ask you which amino acid. So the amino acids, and you can use the abbreviations, well, you may not have it, you can abbreviate by using the first 3 letters. If you're going to abbreviate the code, then you won't have to write as much out, that's fine. And the way you determine that is you use a codon chart. And so you will find codon charts on the internet and image. Okay, so you are going to use this codon chart to figure out which amino acids are being built. So we're not dealing with the anticodons to do this. You're just going to use the RNA codons. So we're starting with A, U, G, and that's pretty easy to do. You're going to start with the big letter for the first letter of the code. So that means you start with the A, and you can narrow that down. And then the middle one was U. Okay, so the middle nitrogen base was a U. And then from there we go to the G, and there's your methionine. So that's how we figure out what's what on the code. So let's try one for U. So this was the anticodon, remember, the RNA code, the messenger RNA code for that, because these wheels will be messenger RNA, is GUC. So we're going to take that GUC to our wheel and narrow it down. And so hopefully you got alanine, yeah? Oh, no, what did I say? Was it GUC? I had it wrong. GUC. So hopefully you didn't get alanine, you got valine. Oh, yeah, all right. Okay, this one's hidden. Great. No, I wanted it hidden. Ah, yeah, this one's hidden. And so, what goes with you? While Harrison's trying to find it, think about what the complement for this was. So, don't get confused when you're working on your worksheet. Just remember that you're going to be using the messenger RNA that you coded directly off of the DNA for this. And that's going to be ACA, right? So, looking at this, give you a chance to sort it out. Is it threonine? Did I get it right? Ooh, yeah. Okay. And sometimes the codon charts look a little different. They'll be in these squares. And so if that's the case, the big letter here is where you would start. So just back to our AUG. Here's our, I can't see. There's the A. And then we come up here. Here's the U's. And then we're going to come over third base to the G. And that takes us to methionine. A, U, and then the bottom row of that. Yippers. Okay, in this moment of role playing, we're going to narrow this up and just say that the cell is the cement area. I don't know what the confused dog is, but she's wondering why Harrison has her carrot. Well, that's not a carrot. That's a virus knife. That's a bad guy. That's a bad guy trying to get in my cell. He's trying to get in my cell. He's a bad guy. I'm not going to let him in. I'm not stupid. Oh, it's not a bad guy. That guy's got keys. I guess he's all right. I don't know. Oh, look, he has a message. He seems like a really nice guy. Oh, he's got a message for me. Oh, he sent me some messenger RNA. Well, you know, I'm just a mild-mannered, hello, my name is, I'm just a mild-mannered ribosomal RNA. Oh, by the way, that was a Christmas ornament a student made when we had a Christmas tree that had organelles all over it. But that's another story. So, you know, I'm just like, I'm back to work. I got my work badge back on, and I'll just go right back to what I was doing as a ribosomal RNA, helping the ribosome say 1, 2, 3, and then I got a double dip here. Transfer RNA would bring that in, and oh wait, Harrison's not. Okay, he's not a virus anymore. He's back to being transfer RNA. I hope you're following this madness, and then would make, and like, I don't know that this is a virus. I'm just a working girl. I'm just doing my job. And we just start making protein after protein after protein. We start making the keys for that virus to put on the outside of its container. We start making the protein container that contains the viruses. And oh, by the way, so there's a protein outer bag, and then there's also a protein, it's kind of spirally shaped, that directly fits the RNA. Dang, and it starts working fast. And once this messenger RNA is in the cytoplasm, it just keeps making anticodon to codon, anticodon to codon. It just keeps making more of itself. It makes more messenger RNA, and it makes more proteins, and dang, it owns you.